Welcome to part 1 of 4. This is chapter 15. In this chapter we're going to be looking at the cell cycle and then our two types of cellular division, which are, the first one is mitosis and then the second one is meiosis. So this first part we're just going to be looking at the cell cycle. The second part is on mitosis, third is on meiosis, and then the fourth part is looking at what happens when chromosomes show different types of variation. So in this chapter we're really focusing on cell division, specifically following chromosomes as the cells go through the division. So every single one of us actually started off with just one cell. So we had the egg from our mother, the sperm cell from the father, there was fertilization, and you started off as one single cell. In order for that one cell to become you, it had to go through many rounds of cell division. And we don't have an exact number on the number of cells that make up a human, but it's close to 10 trillion cells, which is a lot. So this is 10 trillion up there. So that one cell has turned into that many cells, and that makes up an adult human. So very impressive, I might say. So cell division in general is just the reproduction of cells, and this reproduction is very highly regulated. So it's a highly regulated series of events, and we'll get into very small amount of how these events can be regulated, but there's lots of things that the cell has to go through in order to make sure that the DNA is okay, that the cell is dividing properly, so it's under a lot of control. And like I mentioned, there's two types of cell division in eukaryotic cells, so we're going to be looking at mitosis, and we'll also look at meiosis in this chapter. So chapter outline, first off, and I've kind of gone through this already, but we're going to look at the eukaryotic cell cycle, the mitotic cell division, meiosis, and sexual reproduction, and then the fourth part is going to look at variation in chromosomes. So we'll start off with the eukaryotic cell cycle. In the last couple of chapters, we really focused on DNA. And just to remind you, so DNA is a double helix. This is our genetic material. That DNA, it gets associated with different types of proteins, and it folds down into distinct chromosomes, which is way at the bottom. So remember, our DNA, it goes through wrapping. So our DNA is wrapped around proteins called histones to form a nucleosome. Those nucleosomes zigzag back and forth, and that makes up our 30 nanometer fiber. And then we get to our radial loop domains, where the DNA and proteins associate with the nuclear matrix, and that nuclear matrix helps to organize all of these radial loop domains. Those radial loop domains can be condensed down even further until we get to individual chromosomes. So these chromosomes, they occur in sets, and we're going to focus on this from the human perspective. So humans have 23 pairs of chromosomes, or 46 total chromosomes in every single cell. So in every single one of your 10 trillion cells, you have your 23 pairs or 46 total chromosomes. Out of those 23 pairs, 22 of those pairs are said to be autosomes, and then your 23rd pair are your sex chromosomes. And this determines what sex or gender you will become. So you can either be an XX, which codes for female and gives female characteristics, or an XY. If you have a Y chromosome, you'll end up having male characteristics. And in most human cells, because our chromosomes are in these pairs, our cells are said to be diploid or diploid, depending on how you want to pronounce it. So diploid is represented as 2N, so that just means you have 2 copies of every chromosome. So just to kind of relate this back, so humans, we have 23 pairs of chromosomes, so that's our diploid number. When humans get ready to go through reproduction, we form gametes, so the eggs and the sperm. The eggs and the sperm, they have half the number of chromosomes, so they are said to be haploid. So they just have N, so they have just half the number. So our gametes contain one member of each pair of chromosomes, so we're going to have 23 total chromosomes, not the 23 pairs, just 23 total, so half of our number. And this ploidy, the diploid, and the haploid will be really, really important later on when we get to looking at meiosis and sexual reproduction. But I just wanted to introduce those terms right now, so when we get to them, you kind of have a general idea of what I am referring to. And just to show you, so we really focused on humans, but this is a biology class, so we're studying all different forms of life. Just to give you an idea, these are the number of chromosomes that you find in different animals or plants. So goldfish have 100 chromosomes. Your domestic dog has 78. A platypus has 52. Earthworms have 36 total chromosomes. Plants, and we'll get into this more in the last part of this chapter, but plants, there's one plant that has 1,200 chromosomes in every single cell. And I'll kind of go over how a plant can have that many chromosomes in every cell. Dandelions have 8. So you can see that there's a very large range in chromosome number, and it doesn't necessarily correlate with how complex that organism is. So these different pairs of chromosomes, we can actually do something called a karyotype. That is a method where we take a cell that's almost ready to go through division, so all the chromosomes are really condensed down, and we do a cell squash. And then we just take a picture of those chromosomes and use a computer to rearrange those chromosomes based on their size, number, and form. And again, these come from an actively dividing cell, because that's when the chromosomes are more condensed down. So this karyotype is showing a human karyotype. So you can see that each of the chromosomes comes in a pair. So we have our first chromosome in the top left-hand corner, and it's chromosome number 1. And then they just order them based on their size. If you look on the top right-hand side, we have our sex chromosomes. So this individual has 2 X's, so we know it's a female. Another thing about this karyotype, if you look down on chromosome number 21, instead of 2 pairs, this individual has 3 21 chromosomes. So this is a trisomy 21, or generally in the public we know it as Down syndrome. So this individual is a female, and she has Down syndrome because she has that extra 21 chromosome at the bottom. And we'll talk more about these variations in chromosome number, like what we see in this karyotype with chromosome number 21 in the last part of this chapter. So if we just look at those chromosomes, they come in pairs. So remember, we're a diploid species because we have pairs. Members of those pairs of chromosomes are called homologous chromosomes. And they're homologous because they have the same size, the same shape, and they carry similar genes. So they're very similar to each other, and then they pair up with each other. Sex chromosomes, that 23rd pair is a little bit different. If you have 2 X's, they're very similar to each other, but if you have an X and a Y, X and Y's, they differ in size and composition. So the Y chromosome is very, very small. It basically has genes on it that code for different male characteristics. But the X and the Y, we still put them together as a pair. If we focus on just one individual chromosome, so the chromosome up here, it makes this little X that we see in the picture. So this chromosome is actually a replicated chromosome. So each one of those sides is called a chromatid. So you actually have two chromatids that are attached together at the middle, which is called a centromere. And these chromatids, they're identical to each other. So because they're attached, the whole structure is said to be a chromosome. If you pull the two sister chromatids apart, each one of them is considered to be a chromosome. So as long as they're attached, we can say this is one chromosome, but it has sister chromatids on it. Okay, so now we know a little bit about chromosomes. We're going to look through the eukaryotic cell cycle, which is shown here in this diagram. So the eukaryotic cell cycle, it starts off in the light blue interphase part. Specifically, it starts off in the G1, which is the first gap or first growth up here. So the cell up in the upper left-hand corner, this represents a cell that is in the G1 of interphase. So you can see that the chromosomes in this cell, they just have one chromatid on them. So they're just one long singular chromosome. So we don't have those X-shaped chromosomes yet that we were just looking at. So in this G1 phase, what happens in the G1 phase, the cell accumulates molecular changes that cause progression through the cell cycle. So basically in the G1 phase, the cell can grow in size, or it can accumulate different molecular products that it's going to need later on in the cell cycle. In addition, the G1 phase, we have a G1 checkpoint or restriction point. So here's an example of one of those control points. And at this restriction point, or the G1 checkpoint, the cell kind of goes over all of the chromosomes to make sure that the DNA is okay, there's no horrible mutations. And once the DNA is checked at the restriction point, then the cell can pass this point, and the cell is now committed to enter into the S phase, which is the next part in our interphase. So once we pass the G1 phase, then the cell can go into the S phase. And the S phase, you'll notice that our chromosomes, they're really, really squiggly, which means that they're not fully condensed down yet. But you can see that now our chromosomes have the two sister chromatids attached together at the centromere. So now our chromosomes are starting to more look like those X-shaped chromosomes that we looked at a little bit earlier in this lecture. So this S phase, this is where your DNA is replicated, or your chromosomes are replicated, or it stands for DNA synthesis. So after this replication occurs in the S phase, there's two copies of your chromosomes, and they stay connected to each other, they stay joined to each other, and those two copies are called sister chromatids. So the human cell in G1 has 46 chromosomes, and then the same cell after this S phase will have 46 pairs of sister chromatids. And those 46 pairs of sister chromatids, they're still called chromosomes, so we still have 46 chromosomes, but you'll have 92 chromatids in total. Basically what you need to remember is that the S phase is where you have DNA replication occur. And then your chromosomes will now look like this, with the two sister chromatids that are identical to each other, and they're attached at the centromere. And that centromere will be really important when we go through the cellular division part. Once all the DNA has been replicated, the cell enters into the G2 phase of interphase. So G2 phase, this is where the cell synthesizes proteins that it's going to need during the mitosis and cytokinesis events that are coming up. Mitosis, this is where we are going to do our cellular division. Cytokinesis is where the two cells will physically separate from each other. So we have G2, then your cell will go into the M phase, and this is specifically looking at the mitosis phase, but you can also say it's the meiosis phase. Mitosis, we're going to divide it into prophase, prometaphase, metaphase, anaphase, telophase, And then we'll have the cytokinesis that will occur. So this mitotic phase, or the M phase, we're going to look at mitosis. This one cell is going to go through nuclear division, and it will end up as 2 nuclei. And then the cytokinesis is division of the cytoplasm, so the cells will physically separate from each other, and there'll be 2 separate, distinct cells. Cytokinesis doesn't have to always occur, but it usually occurs in most cases of cellular division. So we're going to take our cell. The chromosomes are going to be split in half. So one sister chromatid goes in one direction, the other sister chromatid gets pulled in the opposite direction, and they get pulled apart from each other. So our cells will look more like our original starting cell, where we just have chromosomes that are straight. They're one chromatid. And then the cytokinesis, this is where the 2 cells will physically separate from each other. So in order for a cell to decide if it will divide or not, it can use external factors, so environmental conditions or signaling molecules that come from outside of the cell. An example of this is an environmental condition. If you get a cut on your skin, those cells that were damaged will send out signals to nearby cells that they need to go through division so that they can repair the wound. You can also have internal factors, so there's cell cycle control molecules inside the cell, and then also those checkpoints that I've mentioned before. And we're going to really focus on those checkpoints. So there's 3 critical checkpoints, and we've already actually talked about one that happens in G1. So there's the G1 checkpoint, or that restriction point. That is that point where the cell's going to check all the DNA to make sure that it doesn't have any mutations, that it's okay to replicate the DNA. And then once a cell passes that restriction point, it is committed to basically go through cell division. There's a G2 checkpoint, so this happens in the G2 phase. That occurs after DNA replication. So this is another spot where we can check all the DNA again to make sure there aren't any mutations, and check to make sure that everything was, all the DNA was replicated properly. And then the third checkpoint is the M checkpoint, or the metaphase checkpoint. And I'll point this out later. This occurs during mitosis, and this is where we make sure that our chromosomes are lined up and they're going to be pulled apart properly. And I'll point that metaphase checkpoint out later on when we get to mitosis. Okay, so in this section we talked about the eukaryotic cell cycle, and this is a cell cycle that all cells will go through. So the next section we're going to talk about mitosis, and then we'll get to meiosis and chromosomes.